Hello everyone and welcome to the 7th student. Today we're doing cell and nuclear division, D2.1 in IB biology. So first, cell division. Why is this a thing? All new cells are produced by cell division, okay? That's really important. So a mother cell divides to form two daughter cells. And because all cells come from pre-existing cells, right? This is called the theory of cellular continuity. So this applies to growth, tissue maintenance, and reproduction, all right? So a super important part of cell division is nuclear division, and mitosis and meiosis are both types of nuclear division. Most people think they are cell division. They're not. They are nuclear division. Cell division is wider than that. But first, mitosis produces genetically identical daughter cells, okay? So the diploid chromosome number is maintained. The genome is the same. This is used for growth, repair, and asexual reproduction. Meiosis produces haploid gametes, meaning you go from 2N to N, right? This introduces genetic variation as well through recombination and independent assortment, so it enables evolution through natural selection. There are some shared traits of meiosis and mitosis. So in both of them, DNA must replicate before nuclear division, okay? Also, in both of them, after replication of the DNA, the chromosomes have two identical sister chromatids, which are held together by cohesin proteins. The cohesin loops basically maintain the chromatids together, okay, until anaphase, and then they are cut to allow separation. Also, in both of them, chromosomes are condensed beforehand, okay, and they're also moved throughout the processes. And they're moved, remember, through the microtubules, which form the spindle fibers, okay? Remember, the spindle fibers attached to the kinetochores at centromeres, okay? But we'll talk about that in a second. But just remember the main differences, okay, between mitosis and meiosis, and also the similarities, okay? So then after nuclear division, we have cytokinesis, which is basically dividing the cytoplasm between two daughter cells. So as I said, this happens after nuclear division, important, okay? After mitosis or meiosis. And it ensures that each daughter cell gets cytoplasmic contents, which is super important. It's different in animal and plant cells though, okay? In animal cells, actin and myosin, which are proteins, form a contractile ring at the equator. That's called a cleavage furrow, and that pinches the cell into two. In plant cells, on the other hand, vesicles, which have cellulose, okay, go to the middle of the cell and they form a new cell wall, okay? So this happens in the middle lamella. That's what it's called. And again, these vesicles contain cellulose, which, remember, cellulose is a carbohydrate, right? A polysaccharide, which is structural. So that's why it forms the cell walls. Importantly, cytokinesis is normally equal, meaning both daughter cells receive the same amount of cytoplasm, but it's sometimes unequal, okay? As long as each cell gets a nucleus, they can get different parts of the cytoplasm and survive. So examples are when we create eggs, when females create eggs, there's one large cell, right, that ends up becoming the egg, and the rest are polar bodies that are gotten rid of. And then also yeast budding. So the way yeast divide is through budding, right? A part of it buds and leaves, right? And that's a smaller share of the cytoplasm. So those are two examples to remember. First, we're going to look at mitosis, the phases. I know this is hard for a lot of you. So remember, this is producing genetically identical daughter cells. Okay, so I'm going to give you a little trick to remember this. So first we start with prophase, okay? In prophase, the chromosomes condense, okay, becoming visible. So this is when you can start seeing them under a microscope. Then the spindle fibers form. You can see them here. They start to form. And then the nuclear membrane starts to break down, okay? Then, at metaphase, the chromosomes are going to align at the equator of the cell, all right? And remember, at this point, the sister chromatids are still held together by cohesin, meaning that even though the spindle fibers are already attached to the kinetochores at centromeres, they don't separate because of the cohesin. Then in anaphase, the cohesin is cleaved, meaning that the sister chromatids can be separated. So the kinetochores pull the chromatids to opposite poles through shortening the microtubules. And at this point, okay, importantly, each chromatid is now considered an individual chromosome. So sometimes the terminology can be a bit hard. At this point, they're individual chromosomes. And finally, telophase, how do you remember this? It's the exact opposite to prophase, okay? So here, the chromosomes decondense, the new nuclear membrane forms, okay? And the spindle fibers start to dissolve. So that's a good way to remember. And then cytokinesis happens, which we just looked at, okay? Interphase is the process between each mitosis, okay? Which we're going to look at, the ones that we're doing higher level, we're going to look at that later. So now meiosis, how is it different? So as I mentioned, meiosis produces haploid cells, okay? So that's important. So it actually does two divisions. First, it does the reduction division, okay? The difference with meiosis, which is very important to understand, is that chromosomes pair up. So as you know, we are diploid. So we have two sets of each chromosome. We have two chromosome 1s, two chromosome 22s, two chromosome 23s, etc., right? So in mitosis, this didn't happen. In mitosis, the chromosomes line up individually. But in meiosis, okay, the homologous chromosomes, meaning the two chromosomes for example, attach together, okay? They form a homologous chromosome pair. And at this point, they do what's called crossing over, okay? So crossing over is when the homologous chromosomes exchange DNA. And this provides new combinations of alleles, okay? Because remember, one of your chromosomes comes from your mom, the other from your dad. So by crossing them over, you get new combinations of alleles, okay? That's quite important. Then the homologous chromosomes line up at the equator, like in mitosis. And here's the second part where we get diversity, okay? And this is called random orientation. Basically, it means that out of the two homologous chromosomes, each can go in any given direction. It can go to one side of the cell or the other. And because we have 23 pairs of homologous chromosomes, right, this creates a lot of different combinations for gametes, right? Because depending on which one goes where, we'll end up with different cells in the end. All right, then we're going to have anaphase and telophase like normal. And then we have another division, right? And this other division is very, very similar to mitosis. So at this point, the chromosomes line up individually, right? Not in homologous chromosomes because they've been separated. And then we separate the chromatids, okay? So hopefully that is clear. The result, which is very important to understand, okay, is that we get haploid cells which are all different to one another, right? And we get 4 because we've done 2 divisions. So we get 4 cells, okay? And they're all different. So this is super important for evolution to happen. Great. So now let's look at Down syndrome. Down syndrome, okay, is an example of a non-disjunction. So non-disjunction is an error which happens in meiosis where chromosomes fail to separate properly. So it can happen either in anaphase 1, meaning the homologous chromosomes don't separate well, or in anaphase 2, meaning the sister chromatids fail to separate. This is happening in meiosis, of course, okay? Because for Down syndrome to happen, it needs to be in the germ cell so that it can be passed on to the descendants. Otherwise, it wouldn't have any effect on the progeny. So it can be in anaphase 1 or anaphase 2, okay? And this results in gametes which have either an extra chromosome or a missing chromosome. So then when this abnormal gamete fertilizes with a normal one, it's going to lead to either 1 copy of a chromosome or 3, right? And we only look at Down syndrome, and it's not because it's the only one, okay? This can happen with any chromosome, okay? The thing is, when this happens with most chromosomes, the fetus dies. It's fatal. But Down syndrome is one of the few ones which can result in a living organism. So Down syndrome specifically is a trisomy in chromosome 21, meaning you have 3 chromosome 21s, but it has effects, right? So the kid can have hearing loss, heart defects, vision problems, and also varying degrees of developmental delay. So these are the main things to remember, the things on screen. Okay, so now moving on to higher level. But before we do so, I want to briefly mention that all the slides for all topics, including extensive notes matching each slide, are now available on my website. And if you're still on the fence, you can grab a free sample to check it out. But now let's move on to cell proliferation. So cell proliferation is basically producing new cells. And we do this for 3 main purposes. Growth, right? So when you're going from being a kid to an adult, you have to grow a lot. You need to produce new cells. Cell replacement. So cell replacement, right, can be some cells become old, so then you need to replace them with newer ones. And then tissue repair, right? Tissue repair means, for example, you get a wound, such as in the image, and then to heal the skin, you need to make new cells. All right. So I think it's pretty straightforward, right? But all of these things require mitosis, right? And then reproduction requires meiosis. Good. So this is what I was mentioning before. Mitosis fits into the cell cycle, okay? And the cell cycle is huge. It's way larger than just mitosis. This is the whole process of cell division. So basically, interphase is an active growth phase, okay? Interphase is the whole orange thing, everything else that's not mitosis, basically. And it's an active growth phase between divisions. So what happens here? The DNA decondenses, right? And then the cell doubles in size. So we increase the cytoplasm, the membranes, the enzymes, the ribosomes, right, mitochondria and chloroplasts replicate independently as well because they have their own DNA. Other organelles such as the Golgi can increase by budding, right? But basically we're preparing for division. And this is split into G1, S, and G2. Importantly, S is where the DNA is replicated, okay? Remember, DNA needs to be replicated before mitosis or meiosis can happen, and that happens during the S phase specifically, okay? And then in G2, we continue preparing for mitosis by growing further, okay? But at this point, now we have double the DNA. So, great. And then all of the phases like prophase, metaphase, anaphase, telophase, they all fit into this M, right? So you can see how small it is in the grand scheme of things. G0, importantly, okay, is a step you can go into if you want to exit the cell cycle. Meaning, if you're in G0, you're basically not dividing, okay? You're differentiating. So if a cell doesn't have to divide, which happens very often, then you can go into G0 and just differentiate or not do anything. But it's the way to stop this process from continuing. Good. How is this controlled? So one thing you need to remember, this is not super complicated. You need to know that the cell cycle is regulated by cyclins. So cyclins are these proteins which basically have checkpoints, right? They activate these other things called CDKs, okay? And CDKs trigger phase-specific processes. So cyclins are going to rise and fall, and if they don't reach a threshold, the cell doesn't progress through the cell cycle. That's why it's called a checkpoint. So this ensures that cells only divide when it's needed and at the right time, because otherwise that can be very problematic, as we'll see in a sec. Because what happens when cells divide like crazy? Cancer, okay? So mutations can cause this. Mutations in proto-oncogenes and tumor suppressor genes. So first, proto-oncogenes. Proto-oncogenes are genes that stimulate cell division, okay? So when you mutate those, they become oncogenes, and that can lead to overactive growth. Tumor suppressor genes, on the other hand, do the opposite. They inhibit cell division. But if you mutate them and then they lose their function, you can also have uncontrolled growth. Normally, you need multiple mutations to form tumors, right? But this increases with age, mutagens, and also the amount of cell divisions that happen. Tumors are not all the same, though. We have benign tumors, which means that the cells of the tumor adhere, so they form one mass, and they normally don't spread throughout the body. They're often quite harmless, right? Malignant tumors, on the other hand, are what we call cancer. So the cells detach from each other, they invade tissue, they spread via the blood. That's called metastasis, and they form secondary tumors. These are very, very dangerous, right? So the primary tumor is the original mass, and the secondary is the one that forms after it's spread. And last thing is the mitotic index, which you need to know, which you can just calculate it by dividing the cells in mitosis that you see over the total cells. I doubt this will be in the exam, but it's just here just in case so that you know that it's part of the syllabus. Okay, some questions. So first one, what is the most likely outcome of a loss of function mutation in both copies of a tumor suppressor gene? Okay, 3, 2, and 1. C, okay? If, remember, the tumor suppressor gene stops cell division. If we lose its function, right, if that's the mutation, it's a loss of function in both copies, right, of the gene, we don't have any tumor suppressor gene, meaning we're going to get uncontrolled cell proliferation. The next question is what occurs during the S phase of interphase? Hopefully you remember. Pause now if you want to think, but 3, 2, and 1. The DNA is replicated, right? Remember, G1 and G2 is where the cell grows and replicates the organelles, but S is where the DNA is replicated specifically. Okay. And the last question, what would be the likely effect of a non-functional CDK? Okay. Once again, 3, 2, and 1. Okay. So remember, the CDKs are what the cyclins trigger, and if the cyclins don't reach a threshold, they don't trigger the CDKs, meaning the cell cycle doesn't progress. Meaning if you don't have a CDK rate, the cell cycle is going to arrest at a checkpoint. It doesn't matter if you have cyclins. If the cyclins cannot trigger the CDK, then the cell cycle does not progress, right? It's the opposite to uncontrolled cell division because the cyclins and the CDKs are needed to go through the checkpoints, okay? Great. I think this topic is somewhat easy, but if you have any questions, please do leave them in the comments. And otherwise, I'll see you next week for the next video.